Hi, thank you for joining the webinar. My name is Nicole Suri, and on behalf of MEGIN, I'd like to welcome you all to this MEGIN Masterclass with our guest speakers, Riitta Hari and Matti Hämäläinen, and moderated by Veikko Jousmäki. I'd like to ask attendees to please use the Q&A button on your screen to send in questions at any time during the masterclass, and there will be time for our guests to answer any questions you may have at the end of the session. This webinar will be available to watch on demand on the MEGIN website at a later date. So it gives me great pleasure to hand over to our guests who will officially kick off this webinar. Thank you.
Okay, so we are here at the MEG core of the Aalto neuroimaging infrastructure. So this is an open access infrastructure in functional human neuroimaging. And so we will today have two pioneering people in the field of MEG here presenting their ideas of the past and the future. And I will give the floor to Matti Hämäläinen first, please.
Thank you, Veikko, and thank you, MEGIN, for inviting me here to talk a little bit about the past and the possibilities of the future. So most of this audience is very familiar with MEG, but still it's, I think, useful to remind ourselves that in MEG we measure the brain function by measuring magnetic fields generated by ensembles of neurons. And MEG can precisely tell when things are happening, where they are happening with reasonably good spatial resolution, and also how the brain is connected together. And on the right, we have a picture of primary currents in the brain and associated volume currents. And at the bottom of the slide, there's a kind of a slogan which reminds us that by locating the primary current, we locate the sites and time courses of brain activity. This is about all you need to practically know for the moment about MEG analysis. So in the beginning, when MEG started, there was, of course, an essential ingredient, which is the magnetically fielded room, and we have one of those in here next to us too. And the first magnetically fielded rooms for MEG were built by David Cohen, first in Chicago and then in Boston at MIT. And thanks to the invention of the superconducting squid magnetometer, he was in May 1971 able to measure in real time the human algorithm, as shown here in the strip chart. And on the right, there's a picture of David Cohen himself in the middle. On the right, the inventor of the ARA squid, Jim Zimmerman, and on the left, Ed Edelsack, who was from Office of Naval Research, who funded this pioneering study. And David had a pretty modest prediction in his original paper, saying that I believe the high sensitivity detection system used here shows promise both as a clinical and research tool for studying the brain. When I joined the MEG group here in Otaniemi in 1981, I was certainly inspired by the vision and insight of Olli Lounasmaa, who had the clear idea that we need a whole head MEG instrument to measure all over the brain simultaneously. And in particular, he saw through the difficulties that there's no fundamental difficulty, no fundamental reason why it couldn't be built. And indeed, after about 10 years, in 1992, we built the first whole head MEG device with over 100 channels shown there in the photograph and only under the MEG helmet. And this instrument also marked the start of the commercial operation of Neuromag Oy, which is now called MEGIN. And they distributed many of these instruments and their successors, subsequent instruments, vector views and triaxes all over the world. What enabled this success that Olli was envisioning was that we really were a multidisciplinary team and worked together in the same premises premise is developing instruments, developing analysis methods, and above all doing neuroscience studies with the instruments immediately as they became available. And actually in 1982, Olli on the left hired Riitta Hari, who will speak next, to be the neuroscientist in-house for the MEG team, I would say. And here are a few words which are, I think, essential for the success of this kind of a multidisciplinary research effort, namely that each team member has to be able to contribute somehow to the effort, total effort, and not only contribute, but appreciate the others' contributions. And this requires that you understand more from the contributions of others than a layman would do. So you need to cross the boundaries to the other sides, like, let's say, from physics to neuroscience, and from neuroscience to physics, and to signal processing and estimation theory. And these two first enable each one of us to engage in these activities, and also ultimately abolish the boundaries so that we, in some sense, become exchangeable with each other. And it's interesting that in 1993, when we published the first whole head instrument we built, we wrote that the recordings of magnetic fields over several brain areas simultaneously reveal qualitatively new information on the human brain and open up vast possibilities to investigate the interaction between separate brain structures in evo studies and in the research of brain spontaneous activity. And this was really written in 1993, maybe about 10 years before it became very fashionable in fMRI to talk about spontaneous activity or resting state activity and resting state connectivity. And another component which I haven't yet mentioned is the effort to try to go from signals to the brain. And in this respect, I worked a lot with Risto Ilmoniemi. Both of us are they're shown around 1983 or 1984. And our challenge was really that based on MEG signals, we would try to find where and when the brain is active, that is to find viable solutions to the inverse problem. And ultimately, the methodological development at HUT, Helsinki University of Technology, led to the NeuroMag software used in the prototype instruments, and lastly, even today. But maybe even more importantly, when I was at the MCH for more than 20 years in Boston, the MEG software package was launched and ultimately became the MEG Python package which we know today. And one important aspect of this work was that I became familiar with the computational models of the cortex, which were launched initially by Martti Sereno and Anders Dahle in San Diego, and then Bruce Fischl under his postdoc worked further on the free surface software. And actually now we can very easily use advanced computational modeling in MEG. And so we can, for example, produce figures like this, where we show where connectivity from fusiform phase area, in this case, is different between autism spectrum disorder population and the typically developed population. And I think the open source software has become really important ingredient in MEG. There are many packages now which are quite established. One of them is MEG Python or MEG, which I mentioned, and the other one is Brainstorm. Then there are MEG analysis methods in SPM and so forth. So that these packages together enable people to use the most up-to-date methods. And in fact, as you can see in the picture on the left, these packages are, generally speaking, a group effort. People from different parts of the world contribute to the software, shown here from the MEG coding sprint in 2019, just before COVID. So is there something to look forward to? Certainly, everybody is now talking about optically pumped magnetometers, which work in the room temperature and will enable on-scalp MEG, but they are not yet as easy to use as the split systems, which are kind of foolproof in comparison to the still prototype OPM arrays. And in addition to having the sensor array adapt to the head size, making the measurements closer to the brain, the sensors can be made wearable like EEG electrodes, so that one can study the brain in more naturalistic paradigms than possible with the split systems. And for example, with the OPMs, we can measure everything from babies to the elderly, basically with the same sensors and adapt to the subject's head effectively. So, and something which we cannot really do with splits is to stimulate the brain with TMS and record MEG afterwards, so that it's possible to construct the combined MEG TMS system. And on the right, we see pictures of the TMS MEG system at the MCH, which has 16 TMS coils and 25 OPMs in the same system, and the TMS coils are cooled with the nitrogen, liquid nitrogen system, so that there's a cryocooler on top of the sealed room. And also with OPMs, it's easier to reach new targets like the cerebellum, because we can position the sensors exactly on top of the parts of the brain of interest, and we can make a denser measurement grid, for example, in these parts. And in Boston, with my student John Samuelson, we have studied the possibilities of really making better use of the advanced methods for studying the cerebellum, and have, for example, studied the feasibility of recording something from the cerebellum. And also, we have been able to construct an individualized cortical model of the cerebellum based on just normal 3D MRIs. So, some concluding opinions. I think the MEG is quite special in the sense that the multidisciplinary approach has been the key to advances. This includes instruments, methods, and applications. It's not so clear, for example, in MRI that there would be an immediate, like almost R&D level interaction between the users and engineers and physicists. And the next long sentence describes like a continuous loop where new devices and analytical tools have enabled neuroscience discoveries and clinical applications. And these have in turn led to the construction of new devices and brought about new challenges to the analysis method. So that this kind of interaction is really essential in going forward. But our memories are sometimes very selective, at least. And we tend to forget about what we did in the past and how we succeeded and what kind of stumbling blocks we have had, and really appreciating these past events leads to new breakthroughs faster than just forgetting the past. So finally, there are two sentences in red, and I think in MEG we have been very skilled in being bashful about our achievements. So we have emphasized the non-uniqueness of the inverse problem to the extent that that is probably the only thing that everybody knows about MEG. This is really remarkable. And then another thing which has happened in other fields also, that it's very tempting when you report your results to kind of declare victory too early, so that by doing this you make, for example, the funding agencies conclude that what you are what you have developed is already a done deal. So there's no use to do any research on it anymore. And this is to some extent happening with the OPMs, and maybe we can discuss that later. And just to emphasize that there are many different people needed in multidisciplinary work. There are pictures of many who I worked with, and you know many of them too. There's in the audience, I'm sure. And really, you need to work together daily. You cannot have like a once a month visit to some place and claim that this is collaboration. It's not collaboration. You need to work together daily and at a short distance so that you exchange information. And this is because both information and gossip travel at the speed of light, but information unfortunately travels only 10 meters. And finally, I think for those of us who develop methods and instruments, the challenge is that this what Andy Buckner expressed as, I would love the introduction of a new method be accompanied by a discovery. And Riitta will probably talk more about discoveries now. Thank you.
Okay, good afternoon or morning or evening to everybody. Thanks also from my side for this invitation to talk here. And I have a little bit less scientific presentation than what Matti had. I'm going to tell you how to get hooked for a lifetime by MEG. So in my case, it happened like this, that I was doing my PhD, MD PhD in Helsinki, and I was recording that kind of sustained potentials into long sensory stimuli, like long sounds. And then the problem was that where are these sustained potentials generated? Because in EEG, as these were recorded with EEG, there are problems with electrode skin interface that could cause some fluctuations, and also in eye movements. And then it came from literature, it came an idea that could have quite a strange new method called MEG be of any help here. And why we started thinking that was that there was a publication in two years earlier about eye movements. And people were saying that eye movement artifact, which can be seriously compromise EEG recordings, does not appear to be a major problem in MEG recordings. And this paper was published in EEG Journal 1976. And people, you see here, there's an electrooculogram, electric from left and right ear, not ear, but eye. And then there's eye blinks, which are causing very clear signals here. But in the magnetooculogram, we don't see any deflection like this. So I thought, okay, must be great. We should go towards that direction and record some auditory evoked magnetic fields. And there was also one recording about auditory evoked fields at that time, or at the end of that year, I don't remember exactly. And that people were saying that, okay, so they were recording signals from these 5 locations, getting then these 5 signals. This is 200 milliseconds. And then they went in this direction, and you see that this trace is exactly the same. It's a number 3 here. And they came to the conclusion that evoked magnetic fields appear to be localized to the general area of the primary auditory cortex and diminish rapidly in amplitude as the gradiometer is moved away in any direction. So seemed quite great to try that one, because we knew at that time here at the Helsinki University of Technology, TKK, there was a group working with magnetocardiogram. It was a group by Professor Katila, and they were making measurements in a small non-magnetic sauna building, wooden building, and then all the equipment was kept some 10 meters away. And it was possible to make these recordings only on Wednesday afternoons when the local middle middle wave radio station was not working and causing radio frequencies. So they recorded cardiograms from the babies that were still in the uterus, and then we started to do then some recordings with single channel MEG. And the first results were pretty convincing, I think, because here we have now the electric response, transient 100 millisecond response to long sound, then followed by sustained potential. And in magnetic recordings, we got very similar shapes in the responses, but with polarity reversals between the anterior and posterior parts of the auditory cortex. And then you could model it with some source just in the auditory cortex, as is shown here as well. So now we might ask that, why did we get this kind of signals and polarity reversals, and in the earlier study it was not done? So what went wrong in the earlier study that we so much believed in? First, they had non-optimal recording locations, which is pretty clear because we know that the sources are perpendicular to the cortex. So the source is directed like this, and then you should get signals of different polarities here at this part and this part. But they recorded just on the other side of this dipole, and the same thing here. And also this interstimulus interval that they used, it was 250 milliseconds. It's really short, and then all the meaning that these long latency responses are not clearly visible there. So what we can believe, we can learn from this is just do not just trust earlier results, but check yourself. Maybe they are a good stimulation for your work. And the second thing was, of course, then what about the eye artifacts? That was great news that there will be no eye artifacts, but this turned out not to be the case. So very soon we noticed that if we recorded the eye blinks, so there were huge magnetic signals close to the orbit, and then they decreased when you went back to the head. And in fact, it turns out that because the eyeball is such a very strong static electric dipole, so when it moves, when the eye moves, so there will be very, very big eye artifacts, or when the lid just comes down during the eye blink, so the current can go through this conducting medium and then produce that kind of signals. So that here you see also the field patterns for this eye blinks. So, okay, I got my PhD done, but of course this what I was expecting from MEG was not quite true. So I went back to hospital and got my specialization in clinical neurophysiology, and then also a permanent job there. But then again came something. Somebody showed me MEG, and this time it was the Professor Lounasmaa then who headhunted me to work here in Aalto University, and since then I have been here. So you never thought that, because I had totally different plans. But well, I had to say that this background in clinical and basic neurophysiology has been pivotal for my later work, and maybe for also for the work of many group members. One thing what was very important in this group was that they're great colleagues, students and visitors, and it was really nice to work here. So as you see, people are happy all the time, and it is a really nice thing. You see Veikko, our mentor, mediator today, and then Matti, and then Nina Fors, a clinician, neurologist, and then Olli Lounasmaa, myself. And what was also exciting during these years, that there was so much technical development happening all the time, so that somehow hand to hand with the basic research. So the equipment and these devices, they were developing over the years, and we were excited at each stage, because each time we were able to record something new. First just evoked responses, but then with this whole scalp covering devices, also spontaneous activity. And as Matti was already mentioning, so then from this work also, five of us then founded Mustekala, which is this squid in Finnish, and which then came also, or was at the same time we had a MEG company, which is now after many shifts, it's now MEGIN. And there were, as I mentioned, there were always such a great things in between, although the devices were still small. Like we were really excited in seeing in a single shot polarity reversal of auditory evoked responses. The stimuli were such a like a fricative consonant like H, and then vowel following it. Hey, like word, hey, hi. And then both the beginning of the word and then the beginning of the vowels produced a very clear transient response. And we can see here the polarity reversal. And these were the very low noise squid sensors, and squids we obtained from IBM Claudia Tesse, via Claudia Tesse, so that was important. We were also extremely excited when we got the first whole head covering device, and here's one publication 1 year after the device became available. I still like this a lot because this is an epileptic patient, and you see single spikes, non-average single spikes from right hemisphere superimposed so that all these single spikes are somehow put to the same place. And we see that on the other hemisphere, the spikes are always about 20 milliseconds later. And then you can calculate, for example, the causal delay between these. So it was really quite encouraging. So that we can, if we have accurate information on timing between sources, so it's great and enough if we just know the locations of those sources approximately, not with 0.1 millimeter precision. Over the years, as there was not so many that kind of devices all around the world, we also enjoyed a lot visitors from European countries and also from other countries. But we had a European Union funded Neurobirds large-scale facility where we hosted neuroscientists for altogether 50-person years. And you may see there many prominent MEG researchers like Ole Jensen there, and Bent Lütgenhöne, Joachim Kross, Gabriel Curio, etc. So it was a great stimulating time. And there was, of course, all the time friction also. Many people said that, oh, you don't need MEG. You can do everything with EEG, etc. It's too expensive. It's not a good thing. So you always need mentors and believers who, and with Matti already both mentioned Olli Lounasmaa, who was our mentor, a really important one for the whole work here in whole MEG work here in Finland. Then Ernst Niedermeyer, who has published this EEG Bible, thick, maybe 3 kilograms book. So he invited one chapter about MEG there already in 1993. So that means 30 years ago. So it's really early. And then also Fernando Lopez Silva, he also was really enthusiastic about combining EEG and MEG. So what then, in addition to just recording signals from brain, what has been important? For example, relating brain and peripheral signals has been important. And this is so if you record MEG, and then you at the same time electromyogram from somewhere, it turns out that they are coherent at about 20 Hertz. And for example, here you see that for left hand action or just isometric contraction on the right hemisphere, somatosensory, somatomotor cortex, and for the right hand on the left side. And then later on, there has been other things in addition to this corticomuscular coherence. For example, the peripheral signals have been hand kinematics, like what Veikko has been doing lots of that kind of work, text, corpora, music, etc. There are lots of things to do in that field. And the other thing is that it has turned out that in spite of many skeptical views, it is possible to also use naturalistic setups with live actors, like here we're recording MEG from a person who is just viewing another person making small actions in the hand, and then we see that the primary motor cortex of the viewer is activated. So what can be then said about this bumpy, or this MEG bumpy road? So I would say that this community has been wasting time with so-called exact source locations, which is called magnetic source imaging, the word that I don't like at all, because then the focus goes in a wrong direction, because with MEG we should focus on timing. Also, there has been overselling of single bump solutions, so that people have said that, okay, now we have had this kind of new small evoked response deflection, and it will solve all clinical problems. So people who say like that have probably not been working in clinical environments. And then also, that, as Matti said, that we really need several disciplines to work together to make progress. But many groups have been freezing at multidisciplinary or interdisciplinary stages, not striving towards the convergence research, because if we have unidisciplinary environments, there are people who still say hello to each other, but they have their own games. Then in multidisciplinary groups, they already start talking to each other, but they are just inspired by others. In interdisciplinarity, they start to exchange also their methods, and maybe they change their target problems, but only in so-called convergence research are they really transforming, so that if you look at the colors of the clothing of these people, so they already have been influenced by others, and they have put all their problems to the same basket. So this is like a convergence problem that you need, is especially if you are studying some bigger problems like human brain and mind. And here I would say that only work with people you enjoy working with. And maybe I can say that don't work with assholes, but I just learned from Matti yesterday a new word, that he prefers to work with mass holes from Mass General Hospital. Okay, so important things are that we should really take behavior seriously, much more seriously than has been done. And maybe one should devote at least 1 minute of thinking what a Nobel Prize winner Roger Sperry has said, that the principal function of the nervous system is the coordinated innervation of the musculature. So in evolutionary sense, if we try to understand how the brain works and for what kind of purposes it's really shaped, so we should think that this movement is really important. So it's quite embarrassing how we typically use with just our motor action is something, press a button, don't press a button, and things like that. Then we should also track much more carefully the inhibition, because inhibition is an extremely important part of human brain function, or all brain functions. For example, think about this so-called motor equivalence. So I've been drawing to a tablet these figures with my right index finger, my left index finger, and my right toe, and they all are pretty similar. So that means that if I plan to do something, motor action, so there are several possible outputs available, and the other that I'm not going to use have to be inhibited. And inhibition is really abundant in the brain. And there's been now great work done with looking at the somehow progressions of activation loops in the brain, but we should also pay much more attention to the recurrent connections that exist in the brain, but we should see them between areas and levels. And then more interoception is coming now to all fields of neuroscience. And then also we have with MEG the possibility now to also look better at the deep sources, as Matti was explaining already. And I would really like to see more work in social interaction, where we really look people in those environments where people have evolved. Important is also to combine methods and understand why it's not enough just to combine them, but understand. And for example, MEG and EG seem to weigh the fastest thick pathways, whereas fMRI puts much more emphasis on slow, thin ones. So think about those differences while recording both. And then also we have to remember that MEG and EG are complementary. And so they are like two sides of the same coin, and we should think all the time those both. With INAP-US, we have tried to take that kind of approach in our primer, MEG-EG primer. The second edition should come soon. So about the future, so I think there will be increased availability of MEG devices with these OPMs, for example, improved specification of behavior, I hope, then serious multimodal imaging, open source software and databases for real innovations also, and improved understanding of brain function. That's an important, that's why I put it with red font. And these goals are nicely in line with the timing boom in human brain imaging, because that's also people working with fMRI are getting more and more interested to know also about timing. And just the last comment, that just measuring and analyzing is really fun, but it's not enough. So let's try to go to the next step and really understand the brain. So I thank you, and thank all the funding agencies and colleagues who have made this possible. So thank you. Matti, you will probably, you can, okay, I will go there. Okay, please.
Okay, thank you very much for these excellent talks, and to have a view of the past and view to the future as well. So let me see. So we have a couple of questions here. First question is about what is your favorite discovery in MEG? Who would like to start? Matti, please. Favorite discovery.
In general, my favorite discoveries are these accidental discoveries, which one of which Riitta presented actually a little bit indirectly. This work which started with an investigation of an eye blink field distribution, and then ended up identifying an area which is likely involved in maintaining the integrity of our visual, of our percept when the visual input is interrupted by eye blinks. And this accidental discovery led to actually understanding that some previous monkey literature also applies to humans in the same way. There are some examples like that where you started doing something and then ended up something quite different.
Okay. Yeah, okay, so I still like this early auditory thing, because it taught me so much about this, somehow about the current directions and the relationship between electric and magnetic signals. That was a, and then of course there were also early studies with pain, where we had stimulated that tooth pulp with really painful stimuli, and so it was really nice to see then some local activity in the brain. But then, for example, then this, as I mentioned, this one study which we had these two deflections to words he. So it was really great to do that experiment, because at that time was the first author was inside a magnetically shielded room, and I was measuring, and I saw these two things there, and then I was wondering what it is, and then online I was able to prolong this fricative part, so it was not like it was also with s say, but say, and then I prolonged the first part, and then the second response followed that one, and so it was really a great thing. And maybe I can still mention one. One is also this, like this cortex muscle coherence, because happened like that I was attending a international conference of clinical neurophysiology and just electromyography part, and then I saw that people who were studying just the coherence between different muscles in the hand, they were seeing coherence at 10 hertz, 20 hertz, 40 hertz. Then I somehow understood that there's some similarities to the brain function. And it was also came like out of nowhere. I never thought about that earlier.
So it's worth visiting.
Yeah, conferences.
Visiting a conference for sure. There's another question which is related to the advice to a young scientist aiming at career in MEG. So what should be done? What track should be followed?
Okay. If I start, so that's just like typically we say if the person is going to do some measurements, so then we have these three advices. This look at the data is the first. Second is look at the data, and the third is look at the data. So that Matti,
any comments?
So I would accompany that in the sense that if you look in the dictionary and the word science, it means investigating nature by experiment. So we have somehow forgotten that recent times. I think experiment is still the mother of everything. And then the other one is that usually I refer to myself often that I very accidentally got into MEG. So don't be too worried about what will happen. Don't plan the future because it will be different anyway.
that's true.
So and this happened actually in a building nearby because 5 students were coming to the lab and they were supposed to divide between the MEG project and some physics projects, and we all wanted to go to the MEG project, and then it was we were informed that this won't go, and we still wouldn't back up from our positions, and then we threw dice, and as a result, Jukka Knutila, who now works with MEGIN, and myself were selected to the MEG project, and that's how it was.
Okay, next question. What is the most challenging or interesting MEG research question right now? Maybe cerebellum part, I guess.
That's one interesting thing because now it has been found that cerebellum is important in many more things than we have previously talked about. And when John Samuels and I worked on the cerebellar methods, we were lucky to be in contact with what we call Mr. Cerebellum at MCH, Jeremy Schmarman, who said that finally somebody is interested in the cerebellum. So he was very excited about this. Yeah.
Okay, I already mentioned some of those things like really tracking inhibition, because it's like if you think if you have some external stimuli, they first activate the brain in a certain area, and it said that all activation is inhibitory, then already dies synaptically. So after the synapse, there will be some lateral inhibition or infield like temporal inhibition. So that's one thing. And then also people are doing all kind of very fancy analysis, and then maybe some of the analyses are made just on the whole brain level. But I think one should somehow remember that we have different networks, like sensory networks, and then they change temporally all the time. And in order somehow to be able to look at the networks and their temporal behavior and changes, so that's at least it's challenging enough. And I think it also has functional meaning.
Mm-hmm. Yeah, there was an additional question related to this, but I think this was already answered. Yeah. What do you want to see the field focusing more on? But this is, I think these lines are quite correct.
Yeah, maybe you can add something else, because I had this these recurrent connections. So that's something we know that they exist in the brain, but we don't see typically them in MEG. So we should, they might most likely are working at a little bit different on the basis of monkey data, little bit different frequencies than the feed-forward connections and things like that. But yeah.
you also mentioned this, that for example, fMRI and basically hemodynamic measures and MEG and EEG see different things. We are, I think, very stubbornly stuck with the diagram from last century, where there's temporal resolution and spatial resolution, and different methods are plots in that diagram. And since MEG has a bad spatial resolution and good temporal resolution, it should be combined with fMRI, which has good explicit spatial resolution, without at all taking into account that this is not the temporal and spatial resolution of the same. It's even more striking when you think about PET. PET certainly doesn't have much to do when it maps receptors, for example, with MEG directly, but it supplements them in a very interesting way.
Okay, next question. Do you expect new clinical applications for MEG, conventional or OPM, in the near future?
There's what is a clinical application? So there's maybe a confusion now between clinical research and clinical applications. Clinical research meaning studying patients, for example, to understand more about the disease or understand the healthy brain better through disease. And then there are clinical applications, everyday use of MEG, and maybe in for example pediatric psychiatric diseases there might be some applications of MEG to objectively detect things earlier. But this is a long road from first observation to a clinical application. My primary care physician used this very nice wording, saying that I don't think it's ready for prime time yet. And this prime time is the clinical application, the gold standard, which is very difficult and complicated to reach.
So on the other hand, there's lots of interest now in different type of biomarkers, so that there will be like a combination of features from the measured signal. Then when grouped together, they would predict either somehow the disease or make a improved diagnosis of the disease, or then help in the follow-up. And I think MEG as well as EEG, so they are so rich in this information, because you have so many channels and you have so many frequency bands, then we are now improved computational tools. So I would expect that in that field there will be a clear improvements in the diagnostics at least.
Okay, then we will move on. Wondering any advice for collaborating in a convergent approach? How do I best approach new collaborators both within and outside of my institution?
Yeah,
any practical?
Yeah, I think this is a really important question, and this is a. I think that people have to develop communication. So that's very important. They have to learn to understand each other, and as Matti had something like, they have to contribute, and then they have to also appreciate others. And it's a long way, and there's one good way. So if you find people who are interested in similar questions, then form some kind of just coffee or lunch breaks where you read together a book together and discuss it. And then maybe after 1 year or something, you understand quite well how the other person is thinking. And so this is one way to develop.
Yeah, Matti, any comments?
So I mentioned the distance, and I have sometimes expressed the benchmark for good chances for a collaboration to work, and that is that the parties should be at least at most half an hour away from each other by bicycle.
Yeah, bicycle. Yes, true.
Meaning that you can get to them whenever you have an idea that we should meet. So, and it's still the case that when you are in the phase of let's say, innovating or coming up with new ideas. There's nothing that can beat the one-to-one meeting in person. And the easier it is to do, the easier the collaboration is. And also, another thing is that collaboration is risk. Don't expect every collaboration to succeed. So maybe half of them succeed. That's already quite good. And for example, NIS funds these risky collaborations. You can try for 5 years, and then if it doesn't work very well, it may work to some extent. But if you decide to go apart after 5 years, that's fine. And then you have learned something.
So if I would add something. So in my picture where I have this multidisciplinary and interdisciplinary parts, so I think people are still very egoistic in that phase, because, for example, I would have some problem. I have recorded some MEG signals, and then I know that Matti is very good in developing new software. Then I ask him to help me. So that's still, this will be like interdisciplinary, but it's very egoistic. We don't have the common goals. So one should go one step further, where our problems are somehow we share the problems, and we both are enthusiastic. And this collaboration should start from bottom up, not from that you have heard that, okay, this university will give you now 1 million euros if you will have a physicist and a chemist and a psychologist in the same team. Then you just collect them, and then you come together, and then you go away with when you got the money, and then you work again alone. So it doesn't work like expected.
We have an excellent follow-up question here. Can you both discuss projects that you have been collaborated on together?
Yeah, we've been together.
Yeah, so collaborating.
Yeah, before you went to US, so we had.
But also after that.
Yeah.
Any
Do you remember any?
PubMed.
Yeah, yeah, we have quite many common papers.
Yeah. So really combining the like analyzing the data and
also discussing, and they also discussing all the time the data and also the analysis tools. Somehow somehow not like that he comes with analysis tools and I give it data, and then we have like a window. I give him the data. Please give me the back next Wednesday. So not like this.
So maybe one benchmark or an additional benchmark of this working collaboration is that you are also exchangeable in the sense that you can write a paper together, so that you can sit here like I can type, and then Riitta sees that this is an impossible sentence, and then I give the keyboard to her, then try, and then we converse. There's convergence also in this sense that we start to think in the same way in writing.
And there's one phrase that I remember. Can you explain shouting distance in research? What is the meaning of shouting distance?
So that when you come in the morning, and the computer person has been, which is like a night owl, has been working with the code. You come in the morning, it doesn't work, so you shout, and it's immediately when he or she comes to the, so that they will be immediately then changed. But I would say that in my case, it's been also a little bit problematic, because when I came here from hospital, I left my permanent job in hospital. I came here out of my home base. So there was not very easy ways to go back. So that trapped.
You were trapped here.
Yeah.
Then we have a question related to OPMs. How long before they are of clinical utility?
Well,
so I have some comment now. I have heard 5 years ago it will be 5 years, and this year
I was going to say 5 years. The more diplomatic answer is to say that they are in the horizon.
But I think this whole coverage is something that we need. Or do we need that?
I think we need that. But now there has been a lot of talk that you get better everything when you get closer to the brain, but really the improved spatial resolution is not discussed so much. There, know that NIH is looking at this, what questions could be looked at better when there's a dense local grid of sensors. That's something that cannot be done with squids really.
Then we have another question, a little bit out of the box, so to say. Do you have any funding grant strategies for MEG research? Any suggestions or funding strategies?
So how to apply grants? Prepare the grant early.
Don't go saying.
So it's not a random process. And you don't believe this until you are reviewing yourself. But maybe more seriously, it's related to the early. You should not only leave time in the preparation so that you discuss with the other members of the team what should be in the grant, but also if you can deliberately or by accident get the grant ready and leave it alone for 3 weeks and then have 1 week to finish it, then your brain is kind of refreshed and you can fix it.
Maybe the question was whether you have some suggestions specifically for MEG grants. Are they somehow different from other grants?
No.
Yes. Good science. Good science. Good. Yeah.
They are not different. So let's put it in the other way. So MEG has been accepted as a viable method to study the brain. So if you don't get funded, it's not the fault of MEG. It's the fault of bad science.
Okay, so we are approaching the end of this masterclass. So, do you have any comments at this point? Any greetings or other critics?
Are there any questions?
I think not at the moment.
Yeah,
so
okay.
We have.
There's no further questions in the chat. So if there are no further closing remarks, I would just like to say thank you to our guests for an excellent masterclass, and we've really enjoyed this session, and thank you everyone for attending, and we hope to see you all at future masterclasses. So thank you again.
Thank you.
Thank you.
Bye-bye.
Bye now.
Bye-bye.